Hello, welcome. In this video we will talk about transduction in bacteria. We had already talked about conjugation, transformation. Now we are talking about transduction in bacteria, which is also a type of gene transfer between bacteria. So before we start with what is transduction or transformation, we have to know about the lytic and lysogenic cycle of the bacteriophages, right? So what happens in lytic cycle, that the phage or bacteriophage are viruses. Okay, so these phages are viruses that infects bacteria. Right? So phages are viruses that infects bacteria. So these phage with phage DNA comes and binds to this bacteria. The phage binds to the bacterium, right? Then what happens? The phage DNA enters the host cell. So the phage has a protein coat. So the protein coat stays outside while the DNA is transferred inside the bacterial cell. Then what happens? That the host DNA is digested. This is, we are talking about lytic cycle. So the host DNA is digested and fragmented, like in different fragments. Circular DNA is now fragmented, right? Then what happens? That the phage DNA replicated phage DNA. So here we can see the purple color. The phage DNA was the purple color, so it has replicated. Then the host cell transcribes and translates the phage DNA, producing phage proteins. So the translation and the transcription mechanism of this bacterial cell has been taken up by the viruses, and now virus protein codes and virus DNA are being translated and transcribed, right? So this is new phage molecules are being formed. Then assembly of new phages is complete. A phage encoded enzyme uses the cell to lyse. Then complete phage molecules are produced inside the cell. They are assembled together with DNA and protein codes, etc., etc. And then the phages releases some of the lytic some type of enzymes, that lysis occurs, that the bacterial cell bursts open and these phages come out. Lysis occurs, the new phages are released to start the cycle again. So this is lytic cycle, where the bacteria are killed, right? At the last stage lysis occurs and bacteria are killed. While in the lysogenic cycle, the bacteria does not die, right, until it enters into the lytic cycle. So in lysogenic cycle, similarly, there is this host cell. Then the phage DNA enters into the host cell. Then the phage DNA integrates into the bacterial chromosome and becomes prophage. So the phage DNA entered into the cell and recombination occurred, and here a prophage has been formed. A prophage means bacterial chromosome recombined with the virus or phage DNA, right? So a prophage has been formed, which has an integrated viral DNA within it. So the prophage replicates. This replication continue many cell division. We can see the purple part of these DNA molecules are of viruses of the phage, right, while these are the bacteria. So it had been integrated into the bacterial genome and they are being replicated along with the bacterial genome. Then what happens? The prophage may separate and the cell will enter lytic cycle. So after some time, many cycles of this lysogenic cycle can go on, but after some time, after so much cycle, many cycles, what will happen that the prophage, that this virus DNA will then again get out, right, separate from this bacterial DNA, and then they will enter into the lytic cycle again. The DNA will be ingested, bacterial DNA, and then phage DNA will replicate, and then protein codes and all those things will be generated and assembled, and then lysis of the occur, right. So this is lytic and lysogenic cycle. Now, bacteriophages have two alternative life cycle, lytic and lysogenic, like we studied earlier. In the lytic cycle, a phage attaches to a receptor on the bacterial cell wall and injects DNA into the cell. Inside the host cell, the phage DNA is replicated, transcribed, and translated, producing more phage DNA and phage proteins. New phage particles are assembled from these components, right? Like we saw, this is lytic. We are talking about the lytic cycle actually. So the phage DNA came, its DNA entered. Here its DNA entered, and then the host DNA, the bacterial DNA, is digested. Phage DNA replicated, these purple colored things, and then this host cell started to transcribe and translate phage DNA and producing phage proteins. Then assembled those proteins and the DNAs, and then lysis occurred and the bacterial cell was killed, right. The phage produce enzyme that breaks open the host cell, releasing new phages. These are virulent phages that produce strictly through lytic cycle, always kill the cell. Virulent phages are those phages that always enter the lytic cycle, and lytic cycle of course means that it kills the bacteria. Now here we are, we are already talked about virulent phages. This is about temperate phages, can undergo either lytic or lysogenic cycle, right. The lysogenic cycle begins like lytic cycle, right, but inside the cell the phage DNA integrates into bacterial chromosome where it remains an inactive prophage. So the main difference, it is telling us about the main difference between lytic and lysogenic cycle, that lytic cycle the host DNA is digested and phage particles are released through lysis, but in lysogenic cycle the phage DNA integrates itself into the bacterial chromosome. The purple is integrated into the bacterial chromosome, and then they replicate along with the bacterial chromosome. But after many cycle of lysogenic cycle, they enter into the lytic cycle again. But in lysogenic cycle, not killed, bacteria are not killed, right. So temperate phages can either undergo lytic cycle or may undergo lysogenic cycle. Now we were talking about, this video is about transduction. Of course, the transduction is the process in which a bacterial chromosome is transferred into another bacteria via a phage molecule, right. So virus transfers a bacterial genome to another bacteria, that is transduction. Now there are two types of transduction, generalized transduction and specialized transduction. So in generalized transduction, any gene may be transferred, but in specialized transduction, only a few genes are transferred. So first you need to know that transduction means that a phage molecule, a virus will transfer bacterial DNA into another bacteria, Right? Bacterial DNA will be transferred to another bacteria via a phage DNA. This is known as transduction. So how this happen? In generalized transduction, this is the picture of generalized transduction. So what happens that the phage DNA will enter into the cell when it will be infected. Bacterial cell is infected, so the phage DNA enter into the cell. Then the bacterial chromosome is fragmented. Here the bacterial chromosome is fragmented, and now what happens? The phage protein molecules and the phage DNA is being produced, and some of the bacterial genes become incorporated into few phages. Now that there are in this thing, we can see that there are also, this is the bacterial chromosome fragments, and these are the phage DNA. Purple colored are phage DNA. So what happened that these are the bacterial DNA fragments and these are the phage DNA fragments, and these are phage proteins, right? So some of the bacterial chromosome may enter into this phage protein and become incorporated into it, right? So it is a phage molecule, but it has the DNA of a bacteria, right? Bacterial DNA is present within a phage protein coat, right? So this may occur, and when that occurs and lysis occurred after that, so this phage which has phage protein coat and can infect another bacteria, but has the DNA of a bacteria, right? So this will be transferred, and when this phage will infect another bacteria, it will transfer a bacterial genome. This bacterial genome will be transferred into this bacteria, and of course crossing over will takes place, and a transductant is formed. This is generalized transduction, where any gene may be transferred. While in specialized transduction, what happens that specialized transduction also requires gene transfer of one bacterium to another through phages. Just like generalized transduction, in specialized transduction, gene transfer occurs from one bacterium to another through phages. But here, only genes near particular site of the bacteria chromosomes are transferred. Like we saw earlier in the generalized transduction, any of the fragmented DNA could have been packed and then assembled into the phage and then been transduced. But here only some specialized genes may be transferred. This happens in the lysogenic bacteriophages. In that, generalized transduction occurs in lytic cycle, whereas specialized transduction occur in lysogenic cycles. So what happens in this picture, you can see that the phage is here and it transferred its DNA, the purple colored, and the bacterial chromosomes A plus, B plus, C plus, that is, it can produce whatever protein or amino acid these alleles produces. So these alleles are wild type and can be produced. Now if it undergoes lysogenic cycle, so and then A plus is fragmented and then sent, then this recombination occurs and becomes A plus, B minus and C minus, because this was earlier A minus, B minus, C minus, now it becomes A plus, B minus, C plus, because one of the phage particles had transferred a bacterial chromosome or DNA particle with A plus allele. Second is B plus is being transferred, here C plus is being transferred, and here A plus and B plus are being co-transferred, and here only viral DNA or the phage natural DNA is being transferred, right? So this is a co-transductant where A plus and B plus is being together transferred. The rest are single transductants, where this is non-transductant because here the virus DNA is being transferred. So this is not transduction. Thank you for watching. Like and subscribe the channel.